You know, inside your body, right this very second, there's this incredible, invisible battle going on. And on the front lines, you've got these personal bodyguards, tiny, Y-shaped machines called antibodies. Today, we're going to pull up their blueprint and see exactly how they work to keep you safe.
So have you ever wondered about that? How your body seems to remember a virus it's already fought off? So the next time you encounter it, it's no big deal? Well, the secret to that memory, that power, lies with the heroes of our story today, the antibody. And trust me, by the end of this, you are going to understand their strategy perfectly.
So, where do these bodyguards even operate? You might think they're inside your cells, but nope. They're out in the open waters, patrolling the fluids of your body. And this whole battlefield, it has a very specific name.
It's called humoral immunity. Now, the word humor is a bit of an old-school term, right? It's an ancient word for body fluids, like your blood. So when we say humoral immunity, all we're really talking about is your body's defense system that relies on these special substances just floating around in your bloodstream. And the absolute star players, the special agents of this system, are, you guessed it, the antibodies.
So what exactly are these things made of? Well, they're what we call glycoproteins. That just means they're mostly protein, that's what gives them their core Y shape and structure, but they also have these really critical carbohydrate chains attached to them. It's that combination, the protein and the sugar, that gives them stability and, more importantly, the ability to send out signals to other immune cells.
And what's really fascinating is how we can actually see them in a blood sample. Check this out. When scientists use a technique to separate all the proteins in your blood, they sort themselves into these different bands. And that huge peak right there in what's called the gamma globulin band, bam, that's them. That's the headquarters for your antibodies, or as scientists like to call them, your immunoglobulins.
Okay, so we know what they are and we know where to find them. Now let's get into the really cool part, their structure. We're going to take this antibody apart piece by piece and see how it's just so perfectly designed for its job.
So at its heart, an antibody is built from 4 different protein chains all linked together. The best way to think about it is like a high-tech Y-shaped grappling hook, one that's been precision engineered for a very, very specific mission.
And this 4-chain structure is just a masterpiece of design. You've got 2 big, identical heavy chains and 2 smaller light chains that create the basic frame. But the real genius is how it's split up by function. The very tips of that Y shape are the variable regions. Think of these like custom-made keys, each one designed to fit only a single lock. The stem of the Y, though, that's the constant region. It's like a universal handle that the rest of the immune system instantly recognizes and knows how to grab onto.
Let's zoom in on that blueprint for a second. You can clearly see the heavy and light chains we just talked about, but you can also see how they come together to create even more specialized zones. Notice the Fab region up at the top and the FC region down at the bottom. These 2 parts have completely different but equally vital jobs to do.
This is really where the antibody's split personality shines. The Fab region, that's the 2 arms of the Y, is the specialist. Its one and only job is to find and latch onto one specific enemy, the antigen. But the FC region, the stem, that's the communicator. It doesn't bind to the enemy at all. Instead, once the Fab region grabs on, the FC region basically lights up like a flare, signaling to the rest of the immune system, hey, the target's been marked, come and get it.
And what connects these 2 critical zones? A flexible hinge. Now, this isn't just some simple joint. It's an incredible piece of bioengineering. It allows the antibody's 2 arms to pivot and move around, letting them grab onto targets from all sorts of different angles. This flexibility is also what helps kick off super powerful chain reaction that we're going to talk about in just a minute, the complement cascade.
So we've seen the blueprint, we've taken it apart. Now let's watch this molecular bodyguard actually do its thing. How does this amazing structure take down an intruder? Let's go through the mission plan.
This flowchart right here, this is the entire game plan. It all starts at the very top, the moment an antibody successfully finds and binds to its target antigen. That connection creates what's called an antigen-antibody complex. And once that happens, the antibody has 2 main strategies it can immediately deploy.
Okay, strategy number 1 is to just neutralize the threat on the spot. An antibody can act like a shield, covering up a virus's dangerous parts so it can't infect your cells. Or it can act like a set of handcuffs, clumping a whole bunch of bacteria together. That's called agglutination, which makes them a big, easy target. It can even cause soluble toxins to clump up and basically fall out of your body fluids. In every one of these cases, the enemy is disarmed immediately. But strategy number 2 is to call for backup.
Remember that FC region, the universal handle of the antibody? When an antibody grabs onto an antigen, that FC region actually changes its shape just slightly. And that change activates a whole team of proteins called complement. This is the biological equivalent of sending up a giant flare, signaling to all the heavy hitters of the immune system that a fight is going down right here.
And here's the endgame. All of these paths lead to one thing: total elimination of the threat. This can happen through phagocytosis, where a cleanup crew cell comes along and literally eats the intruder that the antibody has tagged. It can happen through inflammation, which floods the area with even more immune backup. Or, and this is the most direct approach, it can be cell lysis, where the complement system literally punches holes in the enemy cell until it just bursts. It is a total takedown, all orchestrated by that initial antibody.
So we've seen the anatomy, we've seen the action. For this final section, let's tie it all together and really understand why this very specific blueprint is so incredibly important, not just for our bodies, but for the future of medicine.
So let's circle back to that all-important FC region, the handle. Its two most critical jobs are called effector functions. The first is opsonization, which is a great word that basically just means tagging a bad guy to make it look extra tasty to the immune cells that eat intruders. The second is complement fixation, which is the official term for kicking off that whole protein cascade we saw that ends with the enemy cell being destroyed. So yeah, that handle does all the heavy lifting when it comes to signal.
So why does this blueprint matter so much outside of our own bodies? Well, because once you understand how a machine works, you can start to build it yourself. By copying this exact design, scientists have created something incredible called monoclonal antibodies. Imagine taking your body's most elite, most specialized bodyguard and cloning it into an entire army. We can now design these armies to hunt down almost anything, from a specific type of cancer cell to the rogue proteins that cause autoimmune diseases. It has been a total game changer for medicine.
And that brings us right back to the blueprint. We understand the structure, we understand the mission. So the only real question left is, as we get better and better at bioengineering, what new bodyguards are we going to design to fight our future battles? The possibilities are just absolutely amazing. Thanks for joining me.